Welcome to Dr. Balaga's God Knowledge Doc podcast, your quick dive into the latest medical breakthroughs to keep you ahead of the curve. Let's get started. This long podcast will discuss three Nature papers on new insights regarding aging using proteomics. The first, aging isn't one size fits all, how your organs age at different speeds. The second, rapid brain aging occurs at three distinct ages. And the third, feeling suddenly older, scientists see aging waves at ages 44 and 60. Organ aging signatures in the plasma proteome track, health and disease. In this study, the investigators say aging varies across individuals and organs. Organ-specific molecular aging remains poorly understood. Plasma proteomics offer a minimally invasive approach to track organ aging. The study cohort included the longevity cohort. The patients were in it for 15 years, and they evaluated for testing associations between organ aging and risks. 4,979 proteins in 5,676 individuals across 11 major organs were assessed. Machine learning was used to estimate organ-specific biological age. Links were examined between organ aging, diseases, and mortality risk. Key findings: 81.4% of the individuals had accelerated aging in a single organ. 1.7% had multi-organ aging. Accelerated organ aging confers 20 to 50% higher mortality risk, and organ-specific diseases relate to faster aging of those organs. For example, faster heart aging, there was a 250% increased heart failure risk. Brain and vascular aging strongly predicted Alzheimer's disease progression independently from and as strongly as plasma markers, the currently blood-based biomarkers for Alzheimer's disease. Vascular calcification, extracellular matrix alterations, and synaptic protein shedding led to early cognitive decline. Plasma proteomics provided early disease markers and intervention targets. Accelerated aging of the heart was associated with increased risk of heart failure and cardiac arrhythmias. Accelerated aging of the kidneys was linked to diabetes, hypertension, and metabolic disorders. Accelerated aging of the brain correlated with cognitive decline and Alzheimer's disease. Multi-organ aging linked to increased all-cause mortality. This study analyzed aging patterns in 11 major organs using plasma proteomics and machine learning. It found that organ aging varies between individuals within the same body. Accelerated aging in specific organs increased disease risk and mortality. We already talked about the brain and the heart. Accelerated aging in the arteries was associated with vascular calcification and cardiovascular disease. Accelerated aging of the immune system weakened, raising infection and autoimmune risks. Accelerated aging in the liver resulted in metabolic dysfunction and fibrosis risk. Accelerated aging of the lungs reduced lung capacity and respiratory diseases. Accelerated aging of the muscles was associated with sarcopenia and mobility loss. Intestinal aging was associated with altered nutrient absorption and microbiome. Adipose tissue aging affected metabolism and obesity risk. Pancreatic aging was associated with declining insulin regulation and diabetes risk. So key finding was that organ-specific aging is measurable in blood using proteomics and was useful in predicting disease and mortality. Clinical implications: potential for early detection and personalized intervention. Plasma proteomic biomarkers could guide disease prevention strategies. Larger and future studies should validate these studies in diverse populations. In conclusion, organ-specific aging varies widely and impacts disease risk. Plasma proteomics is a powerful tool for tracking aging. Future risks should integrate multiomics and refine clinical applications. The next paper, also in Nature, looked at plasma proteomics to identify biomarkers and changes with brain aging. Study overview, large-scale analysis. There were 10,949 participants using multimodal brain imaging and proteomic profiling. The brain age gap, that is the disparity between chronological age and estimated brain age, was evaluated. Key findings: 13 blood proteins were associated with brain age gap, proteins that were linked to stress, regeneration, and inflammation. Machine learning was used to estimate brain age. Significance: this identified potential biomarkers for brain aging and neurodegenerative diseases, opens avenues for early diagnosis and therapeutic interventions. The key proteins related with brain aging include brevican or BCAN. The lower levels were linked to Alzheimer's disease. These proteins are associated with slower brain aging. The growth differentiation factor 15, GDF15, is associated with faster brain aging. Additional findings: 8 out of the 13 proteins were linked to accelerated aging, and 5 proteins were linked to decelerated aging. Implications: these proteins are potential therapeutic targets for neurodegenerative diseases. It highlights key molecular pathways involved in cognitive decline. These investigators identified 3 peaks in brain aging. At the age of 15 years, there were changes in protein related to metabolism, wound healing, and mental health. At the age of 70 years, changes in proteins involved brain cell function, dementia, and stroke risk. And at the age of 78 years, there were changes in proteins linked to immunity and inflammation. Future directions: potential for early intervention strategies, research needs, validation in diverse populations, exploration of brain-specific protein origins. The third paper, titled Nonlinear Dynamics of Multiomics Profiles During Human Aging, from Michael Snyder's group at Stanford. They investigated molecular changes associated with aging using a multiomics approach. Small cohort of 108 individuals aged from 25 to 75 from California. Median follow-up 1.7 years, maximum 6.8 years. Samples: blood, stool, skin, oral, and nasal swabs. Multi-omics layers were identified. Genomics was evaluated using DNA methylation. Transcriptomics for gene expression. Proteomics and metabolomics using blood biomarkers. And microbiome analysis for gut, skin, oral, and nasal microbiota. They found that age is not a linear process. Molecular changes accelerate at specific inflection points. 2 major transition points were identified. At the age of 44 years, there is increased cardiovascular risk, lipid metabolism and alcohol metabolism is impaired. At the age of 60 years, there is a decline in immune regulation, carbohydrate metabolism shifts, and decline in kidney function. The nonlinear patterns in aging-related pathways included oxidative stress, inflammation and immune function, DNA methylation and histone modification, protein degradation and autophagy. Clinical implications and future directions: early detection of aging-related disease risks, personalized intervention strategies. Future research directions: long-term follow-up studies capturing aging transitions over decades, large diverse cohorts beyond California participants, AI models to predict aging trajectories, Potential applications: aging clocks based on nonlinear biomarker changes, precision medicine for extending health span and preventing diseases. To wrap up, organ aging and plasma proteome tracking. The first study looked at 5,676 adult plasma proteins used to estimate organ-specific aging. 20% of the cohort showed accelerated aging in one organ, 1.7% in multiple organs. Heart, brain, and vascular aging predict Alzheimer's and heart failure risk. Findings support plasma proteomics for disease prediction and organ aging models. The second paper regarding proteomics and brain aging biomarkers was a study of 10,949 healthy adults, and they identified 13 proteins linked to brain age gap. Brevican and GDF15 were most associated with dementia, stroke, and movement disorders. Proteomic changes peak at ages 57, 70, and 78, revealing distinct aging pathways of the brain. And the third paper regarding nonlinear aging patterns, small study, longitudinal study of 108 participants, identified major aging shifts at age of 44 and 60 years. Key pathways affected: at the age of 44, cardiovascular risk, lipid metabolism, and alcohol metabolism were impaired. At the age of 60, immune regulation decline, carbohydrate metabolism shifts, and kidney function decline. In my opinion, that this age identified in the California cohort occurs sooner in Desi people in India, and they are at cardiovascular risk at a younger age. And more people after the age of 60 succumb to COVID, probably due to decline in immune regulation. So these are three important papers in a nutshell. Stand by for more data in this space.